Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biotechnology and Bioengineering at IIT Delhi. Today we are going to talk on the module how and when Ig diversity is created, and this module of course belongs to the paper on immunology. One of the many successes of the clonal selection theory has been to be able to explain how various classes of Ig can be created against the same antigen, because a successful theory has to be able to explain all the phenomenon associated with the antibodies, and this is one feature of the antibody production which we have talked about, that various classes of anti-immunoglobulins or isotypes as we call them, they are created, but their specificity with vis-a-vis antigen remains same. So clonal selection theory eventually was able to explain that, and that was one of the many reasons, of course, why ultimately it gained universal acceptance. We know that initially IgM is produced upon immunization, and then the same B lymphocyte switches over to synthesis of immunoglobulin G. So the objectives of today's module are to understand the meaning of two terms which are used in immunology fairly widely. These terms are allotypes and idiotypes. We also will try to understand that how in various ways this diversity of antibodies is created. We will also understand that how clonal selection theory is very nicely able to explain this phenomenon of class switch. The class, that is the class of the antibody, and the switch means from one class to another with the same specificity. So how this class switch takes place. To explain all these mechanisms of diversity generation and class switching will be within the framework of clonal selection theory. So the concepts basically are involved are very simple. We will devote little bit more time on clonal selection, and then we will explain how this class switching takes place. And of course, class switching would mean that initially you have the production of IgG, IgM, and then the switching over taking place in mostly to IgG or otherwise to IgA, IgD and IgE. We will also learn that how the nature of this class switch to which immunoglobulin ultimately will be produced, that will depend on the site of infection and of course on the nature of the infection, that is the nature of the antigen. The immunoglobulin diversity can be viewed from various perspectives. To start with is a question of having unlimited B repertoire response in terms of generating antibody in response to any antigen. At another level, we have various classes of immunoglobulins produced with specificity towards the same antigen. We have already discussed these various classes. The various classes of immunoglobulins reactive towards a common antigen are called isotypes, and the subclasses of immunoglobulins in a particular class are called subisotypes. So, an immunoglobulin can also be defined in terms of the presence of genetic markers called allotypes. The same immunoglobulin, which would mean that the same isotype against the same antigen in different members of a species, can contain different allotypes. So, these allotypic markers are immunogenic. That is, if an immunoglobulin from one individual is injected to another individual of the same species which lacks that allotype or which has a different allotype, this second individual will produce antibodies directed against the allotypic determinant of the first individual Ig. So, like ABO blood group antigens, allotypic determinants segregate within a species. So, the allotypic variation arises from allelic forms encoded by alleles, which are alternative genes at a single locus. Let us look at the allotypic specificity on IgG by agglutination inhibition. For example, G1M, a locus on IgG1 subtype, an individual can have aspartate, glutamate, leucine, threonine and lysine sequence on IgG G1 molecule, whereas another with a negative allotype will have glutamate, methionine, threonine, lysine sequence instead. In human 25GM, which is a marker on IgG, groups on the gamma heavy chain and 3 on the kappa constant regions are known. Allotypes are also known on other animals such as mouse or rabbit. Allotypes have been recognized by using red cells coated with anti-Rh bearing the GM allotype and agglutinating these cells by mixing with anti-GM antibody. The test sera are evaluated for the presence of the allotype by their capacity to inhibit this agglutination. The allotypes are inherited in a Mendelian fashion. The individuals may be homozygous or heterozygous. The alleles are expressed co-dominantly, so a fraction of Ig of an animal may have one allotype marker and rest of the fraction may have another. An antibody differs from another antibody in its specificity. This specificity arises from the sequence of amino acids in its antigen binding site. Especially hypervariable regions discovered by Kabat's group. Antisera can be raised against an immunoglobulin which are directed against these determinants which characterize its specificity. Such determinants collectively were called idiotypes by Kunkel and Udin. Kunkel and Udin, of course, at that time did not know the existence of or the concept of hypervariable regions. There is a term called private idiotypes. Antibodies raised against an immunoglobulin which react with just one immunoglobulin and none other are said to recognize private idiotype. Some immunoglobulins have closely similar sequences and may share idiotypes. Such idiotypes are called public idiotypes or cross-reacting idiotypes. That is, the antibodies raised against these cross-reacts. Early data on public or private idiotypes were generated in case of anti-dextran immunoglobulin. Anti-idiotypic sera are useful in many applications such as identifying V regions, identifying specific immune complexes in patient serum, identifying VL type amyloid in patient secreting Baines-Jones proteins, detection of residual monoclonal antibody after therapy. After this, let us shift our attention to understand how any immunoglobulin is synthesized like any eukaryotic protein. Its gene also has to be discontinuous, that is, it consists of introns and exons. If a separate gene was responsible for each specific antibody, the nature of IgG structure presented a dilemma. How in each case parts of the L chains and H chains remain constant? How this constancy was maintained in different genes? In 1965, William Dreyer and Claude Bennett had proposed that there are multiple V genes along with a single C gene in the germline. During differentiation, one of the V gene joins the C gene. Dreyer and Bennett's proposal in a way was very bold. It was known that integration of lambda 5 DNA involves splicing of the DNA. Discontinuity of genes for proteins in the case of higher organisms was discovered much later. The hypothesis turned out to be correct. In 1976, Susumu Tonegawa discovered that V and C genes are at a distance in embryonic DNA but are closely associated in the B lymphocyte. It is worth noting that couple of technological developments had to be in place to enable Tonegawa's lab to discover isolation of pure Ig messenger RNA had become possible. Genomic isolation with restriction endonucleases was in place. Hybridization techniques enabled hybridization of messenger RNA specific for constant and variable regions. Thus, Dreyer and Bennett were ahead of their times. Tonegawa's group moved on after this success to try to understand T cell receptor structure. Susumu Tonegawa was awarded Nobel Prize. This finding that Ig chains are coded by multiple genes and by combining V gene with C gene was the first step towards understanding the source of the huge diversity of antibody structures. In an individual, B cells with different antigen specificities are estimated to be in the range of 10 raised to the power 6 to 10 raised to the power 8. So each individual will have these many genes just for antibody production. That is a very significant fraction of the genome. Just by creating Ig with many specificities with a single C gene, reduce this requirement considerably. What ultimately turned out to be the correct picture was slightly more complicated. It was found that mouse have few hundred genes for V kappa and VH. Assuming 300 of each, the combination 300 into 300 would generate 9 into 10 is the power 4 different specificities. From where the extra diversity is coming, as mice is expected much higher number of specific antibodies if needed. This actually related to another old question. Does the diversity of antibody generation have a somatic origin or genetic origin? The information so far indicated that it is genetic in nature, but the potential diversity inherent in the genome did not look adequate. The next insight had to wait determination of the sequence of cloned Ig genes from embryonic and myeloma cells. Many leading labs like those of Tonegawa, Philip Lederer and Leroy Hood collaborated. Leroy Hood later moved out of academia and coined the term system biology, which has now become a frontier area in biological sciences. The first new discovery was that V gene in embryonic cells did not seem to code for entire V region of L and H chains. The germline V gene codes till 95 amino acid residues, whereas V region has 108 amino acids residues. So the next question was, what codes for the 13 amino acid residues? The missing encoding DNA segment turned out to be located near C gene. As it joins V gene and C gene, it was called the J gene. It was found that a number of J genes were there near the C gene in the embryonic cell. J genes have a special significance as they code for the last hypervariable region of the FAB. Hence, they contribute not just to enhancing the number of different antibodies which can be produced, but also directly to creation of diversity of specificities. It was clear that the strategy which seems to be followed by the antibody producing cells during differentiation was that one of the many V genes combine with one of the J gene and then joins the C gene. 250 V genes combining with 5 J gene will generate 1250 diverse complete V gene in the differentiated B cell. This leads to considerable amplification of the diversity of antibody specificities. Hence, the source of antibody diversity ultimately turned out to be both genetic as well as somatic. The genetically diverse pool gets amplified by recombination, which is a somatic process in nature. It may be pointed out that even in fully differentiated B cells, these segments are not joined. that takes place only in antibody producing plasma cells, which is clonally selected by an antigen. In the case of variable region of heavy chains, that is the VH, another kind of gene segment was required in addition to the V segment, J segment and C region. About 15 D segments were found between V segments and J segments. These D for diversity D genes code part of the heavy chains and are sources of further diversity. Just like J segment, these D segments also play an important role in determining the specificity of antibody, which is coded by the complete gene. It may be noted that D segment is responsible for determining the third complementary determining region of the heavy chain. It turned out that the splicing is not always precise. The analysis of amino acid sequence and the corresponding base sequence data indicated that the splicing between the chosen V segment and chosen J segment can be in different ways. This imprecise splicing becomes further source of diversity of variability in both L chains and H chains, as shown in this slide, which illustrates some possibilities. The V gene has CCC sequence, the J gene has TGG sequence. Upon recombination in an imprecise fashion, various possibilities may occur. If the recombination leads to TGG, that means that nothing is contributed by the V gene part of it, then this becomes a codon for tryptophan, or we may get as a result of recombination CGG, which becomes a codon for arginine. Another possibility is that the recombination may result in creation of a CCG codon at the site of recombination, which will lead to the coding for proline. We can also get, of course, CCC remaining with no contribution coming from the J gene, but in that case also this codes for the proline again. An enzyme, terminal deoxyribonucleotidal transferase, is a polymerase which can insert extra nucleotides between VH and D. So this becomes the further source of diversification at the third complementary determining region of the heavy chain. The enzyme is able to do it as though a polymerase, it does not require a template. The enzymes which mediate the site-specific recombination of V, D and J gene segments actually recognize flanking sequences around these segments. The recognition elements are palindromic heptamers with 23 base pairs or 12 base pair spacers. Recombination can occur between sites characterized by 23 base pair and 12 base pair, but not between 23 base pair and 23 base pair, nor with 12 base pair and 12 base pair. V and gene segments are flanked by 23 base pairs. Hence, VV, JJ, VJ recombination cannot take place. This ensures that the sequence which finally is formed has to be a VDJ sequence. Thus, only properly formed IJ gene is expressed. While discussing clonal selection theory, we had talked about allelic exclusion. In a particular cell, only one member of an allelic pair of gene is expressed. Apparently, only in one case the correct recombination takes place, and that is the complete Ig gene which is expressed. This ensures that a single B cell produces Ig with same antigen binding site. The messenger RNA for kappa chain has 1250 bases. It's a typical eukaryotic messenger RNA with untranslated regions at both ends, poly A tail, and a leader sequence. You may recollect that it is this leader sequence which ensures that the corresponding polypeptide chain is secreted out of the cell ultimately. In addition to this, the primary transcript has two introns. During processing of this primary transcript, these introns are removed. Hence, J gene seems to have two kinds of information. First, to combine with V gene, segment at the DNA level itself. Second, to ensure that down the line at messenger RNA transcript level, J segment combines with the C segment. This specific splicing information at two different levels is inherent in the sequence of J gene segment. As far as signal sequence is concerned, the signal sequence of a nascent L chain is shown here. As can be seen, it is rich in the hydrophobic residues. Four domains of H chains are VH, CH1, CH2 and CH3. In addition to this, there is a hinge element between CH1 and CH2. The gene mapping confirms that these five components are encoded by different DNA segments. The hinge region is also very important because it imparts the flexibility to the antibody molecule, so that during the cross-linking between the antigen and antibody molecule, the actual distance between the two antigen binding sites can be appropriately adjusted. We may recall that one of the things which we expected any theory for antibody formation is to be also able to explain that how the different classes of the antibody can be created. This is actually of immense physiological importance because we have seen that in the beginning of the humoral response, the initial immunoglobulin created is IgM, and then the next immunoglobulin which is actually present in the blood in the large amount is IgG. Let us now understand that how this happens and how, if need arises, the same B lymphocyte produces another isotype of immunoglobulin with same antigen binding specificity. This phenomenon is called class switch. This class switch definitely is required, as we mentioned, when B lymphocyte switches to production of IgG after the initial production of IgM. It turns out that the class switch involves only the CH region. The light chain gene is not involved, nor is the VH region. In embryonic mouse cells, CH gene segments of the five classes of Ig lie together next to J gene segments. This includes 4 sub-isotypes of IgG in mouse. So the first to be formed is gene for mu chain, which corresponds to heavy chain of IgM. The further processing, which involves the removal of intron sequence, is done at the primary transcript of messenger RNA level, as illustrated earlier for the L chain. It is understandable that in the class switching, the L chain gene is not involved because all these different classes differ only in the heavy chain. It is also understandable that the VH region is not involved because during class switching, the specificity towards a particular antigen is maintained, and all the classes of the antibodies which are subsequently formed have the same specificity as the initial IgM molecule. The class switch, however, takes place at the DNA level itself. The evidence for this comes from comparison of restriction digest of DNA from both embryonic and melanoma cells. This way, VH DJH remains same, which ensures that antigen binding specificity remains identical. The process can be seen in this figure, where we can see that different parts of the DNA can be looped out. So the recombination takes place at the appropriate point and can lead to the formation of the different classes of immunoglobulins. It may be noted that the change in the CH part, although it does not change the specificity of the immunoglobulin molecule, it changes the other effector functions of the immunoglobulin molecules, and that is understandable because the different classes of the antibodies do differ in terms of their effector functions. Not all vertebrates follow the same mechanism for the generation of diversity of antibodies. In sharks, H chains have repeats of VH, DH1, CH2, JH, CH, and there is no possibility of recombination between different units, hence the diversity is more limited. Chickens have only one VL and one J and one C segment gene. H chains also have only one VH and J segment. They are 15 DH segment but with very similar sequences. How are chickens then able to mount effective immune response? It is found that upstream of a VL gene segments, there are 25 sequences similar to the VL regions, but lack a leader exon and a promoter region. These sections form these pseudogenes can be inserted into the VL regions. This is also a somatic event which is continuously created even after B cells have left bursa. Similarly, about 100 VH pseudogenes produce a diversity for VH segment. So, the origin of diversity seems to be not one but several. To start with, we have a multiple V genes and J genes which already occur. Then we have a multiple possibility of VJ and VDJ recombinations in the case of heavy chains. Then we have a possibility of somatic mutations. In the birds at least, and possibly in other organisms, we also have pseudogenes causing gene conversions. To summarize this lecture, we learnt isotypes, allotypes and idiotypes define the diversity in the structure of immunoglobulins. Antibody diversity arises from multiple copies of V gene, J gene and D gene segments. Somatic recombination of these segments and imprecise splicing generates further diversity. Class switch retains antibody specificity and involves recombination again. For many decades, scientists argued about whether antibody diversity has a genetic origin or somatic origin. As happens in case of many scientific controversies, both groups are partly right. It is both genetic and somatic in nature. Thank you.